Welcome, everyone. We're glad to see you again today for our August EMSL LEARN webinar, where we will be discussing using cryo-electron microscopy for your structural biology research. So for today, let us know where you're coming to us from. We would love to know. So I'm in Richland, Washington, and I understand that there's some folks out there who have been coming to us from around the world. So let us know in the chat where you're coming from. Let's see here. Oh, it says chat is disabled. So sorry to hear that. Okay, well, you know what? We're going to skip that then. I guess the default for chat being disabled was turned on. So we'll get that fixed. So moving on, today for today's webinar, we are going to be discussing using cryo-EM for structural biology research. Make sure next month to join us on September 28th for partner with us, the Molecular Observation Network for Soils, Water, and Aerosol, where we will be discussing Emsel's new decadal initiative called MONET, or the Molecular Observation Network. And you'll learn more about what that's about and how you can get involved. And we'll be sharing the link for you in the chat on how to register for that webinar. Also, just to put a plug in, Emsel's integration meeting is happening on October 4th through the 6th. Abstracts are being accepted until September 2nd for posters and contributed talks, and the registration deadline is September 16th. So please join us for that if you can, and also the registration link is in the chat as well. Today, James Evans is going to provide an overview of the cryo-EM applications for micro ED, and we'll give you some proposal submission tips for requesting time on the cryo-EMs. Amar Parvate will discuss applications of single particle structural analysis of proteins, and Trevor Moser will walk you through applications of electron tomography to visualize cell ultrastructure. We will have a Q&A session after their presentation, so stick around if you have questions. And so with that, James, the floor is yours.
Great, thank you, Linda. So, welcome everyone to our webinar on Emsel's pipeline of cryo-electron microscopy for structural biology. In the talk today, we will focus on 4 major sections. Let's see, slide. There we go. First, I will give an overview of the cryo-EM workflow and microelectron diffraction applications for small molecules and proteins and 3D crystals. Then Amar will introduce single particle imaging of proteins and protein DNA complexes and link to cell-free and native mass spec capabilities that were described in a prior webinar last year. Trevor will then highlight Cryofib Sem and cryo-electron tomography for visualizing cell ultrastructure in situ, so in the native cell environment. And then I'll wrap things up with a short section about things to consider when submitting an access proposal. So with that, let's dive into the cryo-EM workflow and micro-ED. For those of you who may be new to cryo-EM, the technique uses electrons that are accelerated to 100, 200, or 300,000 electron volts. And these electrons then interact with a specimen and form an image on a camera using constructive and destructive interference. 2 critical aspects for cryo-EM methods to be described today is that the real sample is a 3D object, kind of shown here as the red and pink or purple objects that's rotating. And when we collect our images, it's actually the combined additive density projection that we see as a 2D image on our final images collected from the microscope. And as long as we can see that particle from different viewing angles, we can computationally reconstruct the original 3D object. However, this is limited by our ability to see soft biological samples, which are inherently weak phase and amplitude objects and are very sensitive to radiation damage. On the right, you can see a time-lapse movie of microtubules receiving progressively more dose. The microtubules start as fine black lines, and while they're clearly distinguishable at the beginning with high-resolution features, as they get irradiated, the resolution decays, the lines get more blurry, bubbles even begin forming. Those bubbles are actually liberated hydrogen gas that build up into trapped bubbles in the ice from direct damage of the proteins from the electrons that we use to do the imaging. Fortunately, there are low dose methods that allow us to image our biological samples with nanoscale to atomic resolution. And we primarily use 3 different workflows on our cryosat Emsl. The first is small molecule microelectron diffraction. And this is an instance where if you have 3D crystals, nanocrystals of a small molecule or a protein, we can collect continuous rotation diffraction data just like you would at a synchrotron with X-rays. But on our side, we can use very small crystals, collect data sets, and then do direct structure solution with them. Now you'll see here on the bottom of each of the workflows, kind of the typical resolutions that are expected, as well as kind of some caveats or sample requirements. For small molecules, we're easily able to oversample, so we can do direct phasing. But protein crystals, for example, we need to currently work with molecular replacement for phasing. The next approach that we do is single particle imaging. And this is a capability where if you have isolated or purified protein complexes, we can perform single particle analysis where we collect thousands of images, each of which has hundreds of equivalent particles, hopefully in random orientations. And then 2D classification and iterative 3D refinement can then directly solve the 3D structure. Finally, on the far right, you can see cryo-electron tomography or whole cell tomography. And this is used to reconstruct the cellular ultrastructure of whole cells or tissue cross-sections to reveal the native architecture underlying the cellular phenotypes. As mentioned on the far left, our cryosat Emsl is fully commissioned for all of these workflows. And I will give an update on micro-ED capabilities in the next few slides. And as mentioned later on, Amar and Trevor will dive deeper into the single particle and tomography workflows. For now, let's focus on microelectron diffraction. And here, one of the big defining things of our capabilities is that we've taken microelectron diffraction and tried to improve the overall throughput by introducing a new software package we call Auto Micro-ED, which allows us to do near real-time data analysis on micro-ED data collection as it comes off the microscope. Here you can see the overall workflow, but the key features here are that we've built in some steps that allow for interaction with users, and you can turn those interactions off. And so from a single command line call, you can insert one call, hit it, and it will automatically take the micro-ED data sets and provide an ultimate structure if one is found at the end without any interaction. However, you can turn on the interaction points. There's also advanced features that allow you to fine-tune certain parameters and so forth. And so this is actually fully embedded within the Emsl workflow and is standard now for any samples that we do micro-ED with. Down in the bottom left-hand corner, you can see the UPC symbol that you could scan in order to be able to be taken to the GitHub page that has Auto Micro-ED in case you want to install it on your own. In terms of what one of the applications of this technique, especially Auto Micro-ED, it's really helped out with heterogeneity. So to showcase the power, imagine here, actually don't imagine, you can see the results directly. We've intentionally put 2 different crystal types on a grid in order to kind of sample this heterogeneity. These 2 crystals were chosen because they have very similar unit cells in terms of the overall symmetry group as well as overall spacings. And so you can put them on the grid, and they're actually very hard to distinguish on their own. In the upper left corner, you can see the blue squares and red circles that are highlighting the crystals. We actually didn't know those crystal types until we actually processed the data. but you have in a single field of view multiple different crystals, and you can collect data on all of them, send them through the AutoMicroED pipeline. Again, just one command line call, and it'll automatically process all those data sets, separate them into different unit cell groups, emerge the ones that are similar, and provide ultimate structure determination for both of them. And so both of these were actually solved to about 0.6 angstrom resolution with over 80% completeness. This is obviously very exciting, and it's emerging technique, especially for small molecules and proteins. It's really interesting for proteins that are less than 50 kilodaltons in size, which can be really challenging for single particle analysis, or from crystals that are too small for conventional X-ray crystallography, or that have a lot of twinning or other local distortions, meaning you need to sample a very small area in order to get a cohesive crystal and diffraction information. So this kind of ends the brief introduction to AutoMicroED and MicroED workflows at Emsl. Next, I'll hand it over to Amar to highlight our most popular workflow for analyzing purified protein complexes. Amar?
Hi, everyone. My name is Amar. Can everyone hear me? Yes. So I will be talking about the most prolific application of the Titan Cryos, and that is single particle imaging. So very broadly, as James described, this workflow includes collecting high magnification and high resolution images of thousands of copies of the same protein of your interest, followed by extensive image processing, which then gives you a 3D structure with near atomic resolution. So to start with, we purchased a molecule called apoferritin, which is a standard for benchmarking microscopes all over the globe. And we basically loaded the protein on the microscope and collected thousands of images. With the current setup, we can collect images in an automated fashion so that structural biology is not limited by the amount of data collected. These data sets were processed in real time using CryoSpark, and you're looking at a typical interface of CryoSpark on the screen right now, where we have the tasks like CTF correction, motion correction done in real time. So what this allows us is, if the microscope is showing us data that we don't like, we can actually stop it midway, and then we can go back, change parameters, and then the software actually picks up from where we paused it. So here you can see a 2.5 angstrom structure, which is in the rounds of refinement. So basically the CryoSpark Live can take your processing all the way up to refinement. And the take-home message here is that we can go up to structure determination with only minimal intervention from the microscopists involved. So here is a 2D image of the actual protein, which is a ring-like molecule. The software digitally sorts these views into 2D classes, which are then used to obtain a 3D structure of your protein. And here we were able to resolve apoferritin all the way up to 1.9 angstrom. And generally, a sub 2 angstrom structure is considered as a good benchmark for a system like this. So with that, we had another question of what all can we do with this? We know our microscope is benchmarked to a good level. What can we do with this? And then we go to the most infamous virus of our times right now, that's the SARS-CoV-2. I will not be going into too many details about the biology, but it has a protein called NSP15, which basically helps in evading the host immune system. We took this protein provided by our collaborators mentioned on the slide, and then we were able to solve the cryo-EM structure to 2.9 angstrom. Using this structure as a base, we complexed it with another molecule called Exobril-1. Exobril-1 right now is used in anti-Alzheimer research, or rather Alzheimer drug candidate, but it also has some putative qualities or properties against COVID. We were able to obtain a 2.7 angstrom structure of this complex, and right now we're trying to refine it further to see where the Exobril-1 binds so we can actually determine its mode of action. Then we sought to determine how exactly does NSP15 chew up RNA and evade the host system. So we complexed it with an RNA construct, and we obtained a test data set of which we have a 4.5 angstrom structure. This is a work in progress, so we will be collecting more data, and the structure in gray hopefully starts to look like the structure in yellow or blue. There is another capability that I would like to highlight along with cryo-EM, and that is a cell-free protein expression pipeline, which we have on campus. So this basically has you couple your transcription and translation reactions in a test tube. So there are 4 scales of expression, of which I would like to highlight the MIDI, Maxi, and Mega. So the MIDI scale essentially allows you to prep up to 24 proteins in a single day's expression time. There is no purification involved here, but this is a great starting point for some biochemical and functional assays. The Maxi scale allows you to prep up to 6 proteins. The reaction goes on for 2 days, and this amount of protein that you get from this reaction is sufficient for structural studies. Now here we have an expression of a protein called GS, which was produced at all these levels. The biochemistry was performed, and the structure was solved of a protein from the cell-free pipeline. So the next question is, can we solve the protein of our interest produced from this pipeline to, say, a higher resolution? And for that, we turned to, so we turned to a protein called artemin, which is a diapause chaperone found in brine shrimp. So here we use the mega scale cell-free expression system, at which scale we're able to obtain significant and significantly pure sample from the cell-free run, which could directly be used to obtain data from the cryos. The workflow here highlights that while there are 28 days mentioned here, the actual time when we got the protein to where we got the structure is only 9 days. So in less than 2 weeks, we were able to take a DNA construct and take it all the way up to a 2.6 angstrom structure. Later, the structure was solved up to 2 angstrom, but the data collected here, we were able to generate an atomic model de novo. There are 2 things I would like to highlight on this slide. And the first is that we have not only appended one of the most powerful microscopes here at Emsl to other existing capabilities, which the users can take advantage of, we have also in this pipeline mentioned stages at which we can accept samples from the users. We can accept samples as DNA constructs where we can express and purify those proteins here. We can accept purified proteins, or we can also accept grids made by the users. So the users need not know everything about cryo-EM. We can work with them to take it from a DNA all the way up to image processing. On the next slide, we have a link to a YouTube channel where the previous webinar for cell-free expression system is hosted. So the users can check it out after this session is done. And with that, I will be handing it over to my colleague, Trevor, who will be talking about the tomography and some other exciting microscopy we do here at Emsl.
All right, thanks, Amar. Let me get my pointer out here. If I can find it. All right, yeah, so far, you've heard about techniques that generate high resolution structures from protein or small molecule samples that are typically either purified or crystallized. But maybe you have a science question about how a protein complex assembles in its native cellular environment. Or maybe you have a question about how a cell's ultrastructure changes throughout its life cycle, or maybe in response to an environmental stimulus. So for questions like these, which require cellular context, we also do in-situ cryo-electron tomography here at Emsl. So for those of you who are unfamiliar with tomography, the basic premise is that we take our sample, in this case, a bacterial cell, and we tilt it through a range of incrementing tilt angles, collecting 2D projection images at each tilt angle, giving us what we call a tilt series, and that's what you can see here. We can then take that tilt series, we align it, and then back project it to reconstruct the 3-dimensional volume of the sample. So here, we're looking at slices of this reconstructed volume that was generated from this tilt series. Once we have this volume, we can do things like segment out membranes, cell walls, organelles, or other structures in order to build models of the cell. Or in some cases, we can pick out individual protein complexes and determine their structure using a technique called subvolume averaging. In this example here, we've solved the structure of the 70S ribosome from this bacteria to about 9 angstroms using this strategy. And so a couple of considerations for cryo-electron tomography is that we can do this in an automated fashion, similar to single particle analysis and micro-ED, where we can collect tilt series at a rate of around 1 tilt series every 45 minutes. The exact throughput obviously depends on your exact collection strategy for your sample, but that's roughly the throughput we get. And most easily, we can do this on whole bacterial cells, which are natively maximally 750 nanometers in diameter, although the thinner, the better usually. And while this works really nice, as I'm sure all of you are aware, many or most at least biological samples are typically much thicker than 750 nanometers. So that brings us to the Aquelos, which is our Cryofib SEM or cryo dual beam, sometimes they're referred to as. But this is a tool that gives us access to biological samples which are conventionally too thick for TEM imaging without some kind of sectioning. So a quick overview if you're unfamiliar with FibSEMs. Basically, we have 2 imaging columns on the instrument. One of them is an electron beam that we use for scanning electron microscopy, and the other is a gallium ion beam. And we can also use this for imaging, but its main utility is to be used as a milling tool. So this beam of high energy gallium atoms, as it hits the sample, is basically ablating the sample material so that we can use it to cut or section the sample. And these beams are offset from each other. They're not quite exactly orthogonal, but they're pretty close to it, so that if we're milling the sample, the electron beam we're using for imaging is looking down at the freshly milled face. And so there's 2 main functions of the Cryofib, either as a sample preparation tool to make thin sections for TEM, which we'll talk about in upcoming slides. But we can also use it as a data generation tool using a technique we call slice and view imaging. And how this works, I mentioned before, since the beams are offset from each other, this allows us to mill a very thin slice of the sample, generate an SEM image of that freshly milled face, and then mill another sample followed by another SEM image, so on and so forth, until we've effectively created a stack of images of the sample through the volume that we're sectioning. The volume that gets generated can then be segmented and models of the sample can be built. So you can see an example of this here in the video, where you can see this biofilm, this bacterial biofilm, where the bacteria are false colored in orange in this case, and they're combined with some nanoparticles that are false colored in blue. And so this can be really useful for imaging bulk materials like tissues or clumps of cells like we see here. Although while it's good for large volumes, it's generally, well, I should say we're limited in X and Y resolution to a few nanometers, and our Z slice resolution is about 10 nanometers maximally. So maybe you're interested in higher resolution data. And as I mentioned before, there's a couple things we can use a Cryofib for to prepare samples for high resolution TEM. So the first of these techniques we call site-selected lift-out, or sometimes you'll hear to this just referred to as a lift-out. The basic premise here is that if you have a bulk sample that is tens or hundreds of microns in thickness, we can use the Cryofib to physically lift out a much smaller region of interest that we can then take to the TEM to do high resolution imaging on. And so what this looks like is if we have a region of interest, ideally on the top of the bulk sample, in this case, we're interested in the junction between these 2 cells, and we mill most of the material away on either side of our region of interest using the ion beam. So this slab here contains our region of interest. You can see the cell interface at the center of this bridge. We can then bring a cryo-cooled micro-manipulator probe down. We touch it to the sample, weld it together, and then we finish cutting free that slab from the bulk sample. Then when once we cut it free, we can retract it from the microscope or from the bulk sample. And from this image here, you can maybe see why it's commonly referred to as a lift out, as we physically lift the sample out of the bulk material. And once we have the sample lifted out, we can take it to a new TEM grid. We transfer the sample from the microprobe to the TEM grid. We cut the microprobe free, like you can see here, and then we can thin the sample down to a thickness that is amenable for cryo-TEM, which is usually or ideally around 200 nanometers. So while this is a really powerful technique, it is very low throughput. And as you can imagine, it's pretty time intensive to go through all of this milling and welding and lifting out and then further thinning. And it's also fairly risky. There's a lot of opportunities along the way for failure points to happen. And it's not uncommon to have to try several different times in order to get just one lift out. But fortunately, we also have another strategy to do slightly higher throughput of sample generation for cryo-TEM. And we refer to this technique as on-grid lamella thinning. So this is for when you have samples that are usually greater than 1 micron in thickness, but less than probably 20 to 30 microns. But what we do is we tilt the sample so that it's nearly parallel to the ion beam. So we can mill a very thin section that we call a lamella out of the sample. So this is an example here. You can see this movie is playing images from the fib in this case, as we're iteratively taking slices off the top and the bottom of the sample until we get it to a final thickness of, again, usually around 200 nanometers. This other video shows the Sem view of the same sample from the ion beam movie. So here, again, instead of looking parallel to the sample, we're looking down on it from up above. And we use both of these images to track the status of the lamella and to know where we are in the sample as we get it down to its final thickness. So we can usually make many more lamella in a single day of work than we can lift outs. And we can even do this in a semi-automated process sometimes. So the throughput is definitely higher than for lift outs. But for any of these, we'll take the sample into the Cryos to do TEM. Just as an example here, you can see a low magnification images of this lamella where you can see it's opaque where it's too thick on the sides, and we are electron transparent in the lamella region where it's been thinned down. And then corresponding here, there's a couple of high magnification images where you can start to see the periodicity of this protein crystal that we have thinned. And I do want to point out that in this example, we've thinned down a crystal that is normally would be too thick to do micro-ED on. So it's several microns in thickness natively. But now that we've thinned it down to 200 nanometers, it's now thin enough for micro-ED. So this opens up a lot of interesting opportunities and flexibility for samples to be used for micro-ED. We can, of course, still take micro or nano crystals that are natively thin enough, but if your crystal size is a limitation and they're a bit bigger, we can still get access to them by thinning in the Cryofib first. And so crystals are one application for on-grid lamella thinning. Another, of course, as you might imagine, is for cells that are conventionally too thick to image without sectioning. So in this example here, you can see these chains of cyanobacteria. And normally or natively, these are a couple microns in diameter, so too thick to transmit through without sectioning. And similar to the last slide, you can see our SEM and ion beam movies as we're thinning the sample down to this final lamella of 200 nanometers in thickness. This is what the resulting lamella looks like in the TEM. So you can see our nice single cell chain of these cyanobacteria kind of connected together here. And in this case, for non-crystalline samples, instead of doing micro-ED, we can do electron tomography of the resulting lamella, similar to how I talked about in the first slide. So here you can see the 3D reconstruction of these cells, or at least the interface between two of these cyanobacterial cells. And similar to conventional electron tomography, as I mentioned earlier, we can also pull out features of interest. So obviously there's a lot going on in these particular samples, but one thing in particular we're interested about, maybe you might be hard to see in the large field of view, but we have a blown up region of these gap junctions that are spanning this intercellular space. So this is something we're working on collecting some more data for in order to pull these out to do subvolume averaging for these structures. All right, and so finally, I just wanted to touch on data collection and processing. And as with most current cryo-EM methods, the name of the game is automation and throughput. Usually we require large volumes of data in order to solve structures. So while a lot of the methodology for single particle analysis, for example, is pretty well established, cryo-electron tomography is still, at least currently, a little bit less routine. So we spent a lot of time either partially or fully automating as many of the steps of our pipeline that we can. So sample preparation is still largely hands-on, although as I mentioned, there are some parts of lamella thinning that can be automated, although we find this to be highly sample dependent so far. Although once we have our samples frozen, data collection is pretty much fully automated. And as I mentioned before, we can usually get throughput around 1 tilt series every 45 minutes. Potentially we can go faster as needed, and this is something that we're working on improving the throughput on as well to get this to a faster speed. For data processing then, so if you're already familiar with data processing for electron tomography, we can of course provide raw data if that's all you're looking for. But if you're just getting started in the field, we additionally have some scripts that can do automated pre-processing of the data, followed by tilt series alignment and reconstruction. And that we can make that available to users as well if you're interested in getting pre-processed data. For downstream processing, again, for things like segmentation or subvolume averaging, mostly this is still currently manual, something we're working on improving the throughput for as well, but that's something we can help as well with if you're interested in it. So hopefully I was able to demonstrate how in situ cryo-electron tomography can be used to determine biological structures in their native environment, importantly, and especially how our Oculos really expands the range of biological samples that we have access to. As I mentioned, there's quite a few different techniques I've sort of covered briefly here that are available, in which exact one you might want to use is generally sample dependent. But if you're interested, we're happy to help out and discuss what might work best for your particular sample and science question. And so with that, I'll kick it back over to James, who will talk to you a bit more about methods for getting access.
Thanks, Trevor. So now you've heard about the various workflows for imaging samples with cryo-EM at Emsl. The next few slides, I'll try to detail ways that you can gain access to these resources, and also a few tips about how to tailor your particular proposal to try and increase the chance of it getting awarded. So there are two main ways to get approved access to the Cryos at Emsl. Since the microscope and its operations are co-funded by both Emsl and Department of Energy, BER, Biological and Environmental Research, Division of BSSD, they each have 50% access to the instrument. The BSSD access mechanism allows cryo-EM only access, meaning you can combine access to the Cryos, Oculos, and Tahoma for cryo-EM related research on any of our workflows that we presented today. But you can't access other equipment with this mechanism. So to combine cryo-EM access with any other Emsl capability, like the cell-free expression system, super resolution fluorescence, native mass spectrometry, NMR, or other capability on site at Emsl, you need to apply for a standard Emsl proposal. Fortunately, there's many different types of Emsl proposals, and this page shows the link to the web page at the very top that takes you to what you see in the scrolling movie. This page gets updated quite frequently with the different types of proposals that are available through Emsl, the scope of work, the scale of experiments, the time frame that you can access the resources if awarded, as well as critically the due dates for them. The bottom of the page links take you to the specific Cryos and Oculos web pages, so you can learn more information about those capabilities that we've described today, as well as links to other synergistic instruments that you could potentially take advantage of. It also lists points of contact that you can reach out to in order to ask specific questions regarding your particular samples of interest. And we are very happy to help in the formulation of your proposals to help boost technical feasibility. And so the website link at the very top is really helpful for finding out information of the different types of proposals for Emsl, and the links at the bottom are really helpful for getting in contact with instrument custodians and so forth in order to start your journey here. However, as mentioned earlier, there is the second and separate mechanism for accessing the Cryos and Oculos for only cryo-EM projects. And this uses a special access mechanism through Emsl still. It's called a resource owner proposal. And in order to get a copy of the template and instructions on how to submit, please reach out to me directly. We unfortunately don't have this information listed on our website because it is a special access mechanism. But regardless of how you access the cryo-EM workflow, if your proposal is awarded, you wind up gaining access to not only the state-of-the-art instrumentation described here, as well as access to our expert staff who will do most of the imaging and help shuttle your project to completion, as well as access to image processing resources, things that we provide, immediate processing that we provide, as well as additional resources that you could take advantage of to continue processing them. And all of this access to the instrumentation, staff, and image processing are available to you free of charge through these user proposals. So with that, we are excited to help you advance your biological and environmental research using cryo-EM. And to finish, I'd just like to emphasize the overall team. Sorry, there's a lag in changing the slides apparently. There we go. So our team is shown here. In addition to Trevor and Amar, who joined me on this webinar, the other two core members of our team are Irina Novikova and Samantha Powell, who are involved with cell-free expression, single particle analysis, microelectron diffraction, and the interface with mass spectrometry. Irina and Sam were critical, along with Dunam, on the portion of the automicro-ED process that I described in the beginning of the talk. In addition, we also draw expertise from others within PNNL in order to address needs that come up through external collaborators, internal research, as well as overall user science. So I'd like to thank PNNL collaborators Jory, Dunam, and Moe, as well as the external collaborators whose projects, we didn't really go into the science in them in this talk, but helped to illuminate the overall methods and workflows. So Michelle Lieberton, Himadri Prakrasi, Michael Knobloch, Jennifer Hyde, and Wes Voorhees. And finally, I'd really like to acknowledge the funding both from Emsl as well as from DOE BER for both the capital purchase of the Krios as well as the longer term operations of it to provide access to the general science community. And with that, we are done and happy to take any questions.
All right. Well, thank you. That was a really great presentations that you guys gave. So we did get a few questions from the audience that we'd like to start out with. Let's see. So we had a couple anonymous folks attending, so I'll start with them. Can you feed tissues, a few layers of cells, into your micro-ED pipeline? If not readily, is it still doable with some tweaks? Input would be plant cell leaf with inclusions and crystals. And I believe that one was for you, James.
Yeah, so I partially answered it in the Q&A. So it definitely is possible. Trevor actually provided a slide on this, and it looked like the anonymous attendee who sent that question noticed that at the same time I sent the chat. So in principle, you could take plant tissue, any type of tissue of whatever size and so forth. The thickness of the tissue will dictate how we try and preserve the overall sample, but you can take that sample and then get it into the aquilose and do either lamella thinning if you're able to target your particular area of interest. So a crystal inside of the plant cells or an inclusion. If we can target that, then yes, we could do cross-sectional milling in order to create a thin enough lamella. Or if it's already exposed at the surface, then lift out becomes available. So Trevor gave a great example of that already. The biggest thing for throughput is going to be whether or not you can really target and whether or not you can, let's put this another way, the overall thickness of the sample will dictate things. So hopefully that addressed the question.
All right. Yes, it was a repeat. I just wanted to make sure that other members of the audience were able to hear it and hear the response as well. Another anonymous submission, can single particle analyses be applied to resolve the structure of proteins when assembled on substrate/mineral surfaces? And what are the challenges? And that question was directed to Trevor.
I guess the short answer is it probably depends. If you're adhered to a substrate, it still needs to be electron transparent. So if you're adhered to a substrate that is microns in thickness, there will be problems. But if maybe you have very small nanoparticles that are associated, or nanoclusters of, metal atoms or something that your protein is in complex with, that becomes more doable. The other challenge, I guess, that goes in with that is sample heterogeneity is usually one of the major limitations of single particle, because we're averaging thousands of particles together, tens of thousands of particles together. If you have, for example, samples that are have different conformational binding points to whatever substrate you have, or maybe some are bound, some are not bound, that can impact and limit what your final resolution will be. But so again, it kind of depends on exactly what your substrate is that you're binding to, but potentially it's doable.
Okay. So the
Can
next
I add one more thing to that? So Trevor's answer is perfect. And to deal with heterogeneity, if the protein complex that's interacting with the mineral is big enough, tomography and subtomogram averaging is also possible, and that does partially get around the heterogeneity issue of conformational states. So it really depends on the overall sample.
Okay. So Dunam Kim asked, typically for subtomogram tomography averaging, so STA, how many subvolumes are averaged for higher resolution? Hundreds of them? And that was addressed to anybody.
Sure. So the answer again is it depends. And really what it depends on is what your target resolution is. And the general rule of thumb is the more particles you are averaging, the better your signal to noise and the higher final resolution you can achieve. So that's one of many factors. But so you'll see anything, people, there's been structures published with thousands of particles averaged together in order to generate structures, and some as few as hundreds. Obviously, there's a differing in results that you'll get out of those, but somewhere hundreds to thousands of particles is typical for subvolume averaging.
Okay. So Zina, I hope I pronounced your name right, was asking, so just curious, is there a specific sample introduction system that is used prior to running the sample to perform single particle analysis as a means of separation of proteins? And that question is directed to Amar.
So the short answer is yes. Typically, we would like to have a monodisperse and fairly pure sample when we load it on the microscope. However, biochemistry in and itself is challenging, and we are aware of that. But typically, we would prefer to have the sample run over a size exclusion column so that we can obtain a very pure fraction and then load it on the microscope. And as an addendum to that, if we get a sample here that after imaging we realize is not as monodisperse and as homogeneous as we want, then we have in-house size exclusion chromatogram machines like FPLCs that we can use to clean it up and then rerun the sample, reload the sample on the microscope. So we have the kind of protein distribution that gives us high resolution structures.
Okay. So we have another question to anybody on the panel, and this is from another anonymous attendee. Will the single particle imaging be able to reveal the molecular structure, e.g. the cytochrome C, in a intact bacterial cell nanoparticle conjugate? Do you want me to repeat that?
I could attempt to answer that one.
Okay.
So I'd assume in this case that the cytochrome C is what the nanoparticle is conjugated to and so forth for this answer. So if that is the case, then it really depends on copy number, and you wouldn't be using single particle in that case. You would most likely be doing electron tomography on that. And so if you do have that and the interaction or conjugation between cytochrome C and the nanoparticle is fairly confined, it's either a rigid interaction or very narrow distance, then you limit the flexibility of where the nanoparticle is relative to the cytochrome C. And so if you have kind of a similar location for that, then you could potentially do tomography data collection, box out all of the areas where you have the nanoparticle conjugated to cytochrome C, and then do subtomogram averaging of it. And as long as there's not a lot of flexibility, then you would presumably be able to do it. However, subtomogram averaging is highly dependent on overall protein molecular weight. So because of the thickness of the sample for tomography, that degrades the overall contrast that you see. And so small proteins, sometimes you can't even detect those in tomographs just because there's not enough signal to be able to distinguish them. So the answer comes down to there is feasibility. You would want to have a rigid linking between that conjugation, and then it comes down to overall protein size. But there's potential ways to be able to do that. If, like was mentioned earlier, you can easily target those. You can have cells that are pre-coated with nanoparticles. If they're only binding to cytochrome C, put those into the aquilose, create a very, very thin cross-section that has some of your conjugated protein with nanoparticle in it. And then that will increase your signal to noise and increase the chance of being able to see the small proteins. But it also decreases your throughput because you have fewer copy numbers to average together per data set that's collected. So, yeah. Trevor, do you want to add anything?
No, I think you hit it.
Okay. All right, next question. Can cryo-Fib SEM be performed on whole cells that have been tagged with something like a nano gold particle, or is it incompatible?
I guess I can take that one. Yeah, so absolutely we can. So yeah, I think if I understand right, you've got nanoparticles which are somehow tagged to cells which have been frozen. We can absolutely do Cryofib on those samples, and that is actually maybe useful if you're trying to do something like targeting. You need to know where to cut your lamella or where to cut your lift out. That is, if you have some high contrast fiducial that is attached to your feature of interest, then that definitely aids with the milling process. So yes, that should absolutely be compatible.
Okay. So the next one is from Dharani. Is it possible to analyze proteins assembled at the interface of micron scale liquid droplets using cryo-EM? And if it's doable, what are the associated challenges? Open to anybody. Do you want me to?
So, Amar, do you want to take that one, or?
I forgot I was not unmuted. Right. Right. So yes, we can look at proteins assembled at the interface of liquid droplets, with the caveat being, what droplets are we talking about? If the droplets are something like oil or something that's electron opaque, then it's going to be challenging. However, we're not ruling that out. The challenges can be, we often run into proteins having a preferred orientation at interfaces. So as James mentioned at the beginning of the webinar, that we would prefer to have proteins in all sorts of orientations, so we get their 3D views. In this case, we may not get those 3D views. So after some initial tests, we will have to reevaluate whether a single particle is the way to go forward, or whether we want to maybe switch to single subtomogram averaging. But yes, we can perform cryo-EM on micron-sized liquid droplets. James, if you want to add something to that.
The only other thing that I would say to that would be, if it is a micron scale one, because that's generally too thick, you would ideally want to freeze them in solution, and then we would put them into the aqueous to do lamella thinning. And so, yes, you could do the same thing, treat it like it was a cell, because of the scale of those liquid droplets and so forth. That would be the only thing to add to what Amar said.
Okay. So a follow-up question, typically how many hours are required for sub-tomogram computation? And that's directed to Trevor or anybody really.
Yeah, I guess again, depends on how many particles that you're trying to average together. And it also depends on how difficult your particles are to pick out of the volumes. So if your particles are readily identifiable, and you can fairly easily mark their positions for extraction, and let's say you've got a few hundred particles, probably assuming everything went perfectly through your pipeline, you could probably have a structure within 24 to 36 hours, I would think, computationally. And I'm being a little bit generous to be safe on that. Again, usually there's lots of things along the way that interrupt that, but I think minimally that's probably what you're looking at for computation time.
Okay. So Vancy is asking a more general cryo question. When do you expect researchers to start using SPA high-res structures to search through tomograms to find their proteins of interest in situ? And then is it a computational restriction right now, or is it the unavailability of enough tomograms to search through?
I can answer that. So we don't so being able to mine a tomogram to try and look at location of a particular protein of interest, interactions, and so forth, that's technically feasible right now. You don't need to use single particle analysis and purify your protein of interest to get the structure of it, just to be able to do mining of a tomography data set to find it in its location. So many proteins have their structure already determined. AlphaFold and Rosetta Fold have created a huge database of available models and so forth. So if a model is already available, that's pretty good match and pretty high confidence for your particular protein of interest, you can actually use that as a template right now in order to do template matching and mining of your tomography data. So that is feasible. It does have challenges, like we mentioned, based off of particle size. Subtomogram averaging in general has been very successful for things over about 50 kilodaltons or bigger, significantly bigger, very similar to single particle. Things considerably smaller than that are very difficult to do. So I would say that the critical threshold right now is really dependent on what particular protein you're interested in, what the size of it is, and that will kind of dictate whether or not it's really feasible. If it is feasible, then pretty much the next question becomes copy number. If it is a very high copy number, that protein should exist in like 1,000 of them per cell and so forth, and they're readily available to identify and so forth, then your restriction becomes more on collecting enough data sets to be able to capture enough to do subtomogram averaging. If your copy number is very low, then that becomes even more so. Just getting the data sets enough to be able to interpret appropriately is the issue. So hopefully that addressed the question.
Okay, another question from Zina. When performing tomography only for slicing purposes, can generated lamellas be used for other analyses afterwards, like elemental mapping?
Yeah, that's it. That's an interesting question. I don't see any reason why you couldn't depend. I think I'm going to guess your elemental mapping question is related to electron spectroscopy. That would be the easiest, considering we have electron, or usually the samples are made for electron microscopy already anyways. The only considerations I would caution against is that if you've already collected data on your sample, that it has been damaged to some extent by the electron beam already. If you wanted to go to another elemental analysis, I'm not sure, like SIMS or something like this. I'm not sure that's something we've done here, but you would have to just make sure that you can do that transfer, keep it under cryo conditions while you're doing your elemental analysis. But otherwise, if you can do that, I don't see a reason why you couldn't.
Okay. We have 5 more minutes left for questions. So if anybody in the audience still has a question, please let us know now. And we have a couple more that we can run through. How does Emsl distribute data that's collected to users? And again, anyone in the panel.
Since nobody else is talking, I guess I can take it. All raw data and some of the process data is made available to users through the MyEmsl archive, which is available through the user portal. So when you get an approved project, you go in through the user portal and you can access your data directly from there. The MyEmsl download link is a web-based application, but for a lot of the all of the electron microscopy data sets that we collect on the Krios and Oculos, those data sets are fairly large. We're talking upwards of a couple terabytes in many cases. And so we also provide links for direct download with wget or another parallel file transfer program. So all raw data and most process data is the short version.
Okay. Are there Emsl computing resources for processing cryo-EM data once collected, or do users have to do that on their own end?
I guess I can take that. So for most projects, we do some initial data processing, and we provide both the raw data and the process data files to the users. Users can request additional computational resources. One of them is Tahoma, to which users can get access. So yeah, the users do not have to do all the data processing at their end. We do offer assistance with that.
Great. We have time for 1 or 2 more questions. So if I have samples that are already frozen and screened for cryo-EM data collection, how can I get them to Emsl?
So we have a cryogenic dry shipper that we use commonly if our users have samples and they're trying to get them to us frozen. We have this dry shipper that we can send out to users. They put their samples in a cold state and then ship it back to us. And we find this usually does a pretty good job of keeping samples cryogenically preserved over shipping across the country even. So.
All right. Is there an ongoing collaboration, or in an ongoing collaboration, do I get to go back and send modified or more samples?
So again, I can take this. So yeah, we will provide direct feedback on imaging sessions to the collaborators. We will also consult with them if more or modified samples are needed, or if some biochemistry needs to be changed just to get better data and better results. So the users don't need to have the perfect sample going in. The users typically do everything they can at their end, but again, if we find that something needs to be changed, we will work with the collaborators to make the samples more amenable for high resolution work. Okay.
You guys are answering these questions very efficiently. So we do have a little bit, just another minute. Does Emsl provide any training opportunities or workshops for cryo-EM?
Yeah, I can take that. So right now we do not offer any workshops, though we are internally discussing and formulating plans for what this would look like in the near future. So I'd say stay tuned for workshops. However, if you have an approved project, users can come on site and be trained on various aspects of the workflow and shadow our microscope operators in a one-on-one format to get familiar with the process overall and so forth, or to kind of help steer experiments.
Wonderful. All right, well, we are done for today. I just thank you everyone for attending, and thank you to the panelists. Your presentations were very informative. In the chat, if anybody's interested, our September 28th webinar for the Molecular Observation Network, the registration link is available there. So please sign up. That's going to be September 28th again on our Molecular Observation Network. Lots of opportunities to get involved in that effort. And also don't forget, Emsl integration is happening on October 4th through 6th, which is going to focus on artificial intelligence and machine learning for biological and environmental research. So if you're interested in AI, ML, that's definitely a conference that you'll want to attend. And registration deadline for that is September 16th. So with that, thank you everyone. Have a great rest of your day. Bye. See you next month.